Hi, Lex. Thanks for joining me. So today I wanted to talk with you about the recent research from Dr. Puidley's lab in Boston and how that might relate to the ADA2 now meter that the foundation has been developing for a few years. So first, can you just give us a summary of Dr. Lee's study?
Well, the first and most important thing is that the full body of Dr. Lee's work really strengthens the argument that ADA2 is a practical biomarker that could help us with a number of things. First, it could help with the screening of undiagnosed patients. For example, siblings or other close blood relatives of a patient diagnosed with ADA2 could be very quickly screened with a biomarker such as ADA2 blood enzyme levels. And we could assess their situation and, if necessary, start treatment almost immediately.
So does this biomarker help in ways that are beyond just a diagnosis?
Yes, biomarkers are important because first, they give real-time information about what's happening in the body. And second, genetic tests don't really tell you how the mutation is affecting patient health. A biomarker, like an enzyme activity, can show whether the enzyme is actually working, how severe the deficiency is, and in the future, measurement of ADA2 as a biomarker can help monitor treatment outcomes. For example, if a patient receives a gene therapy, it's going to be critical to know that the gene therapy has successfully restored the enzyme activity in the blood. So genetics equals potential for disease, whereas biomarker equals the reality.
So how does the cost of genetic screening versus an enzyme activity test compare? And then what are the implications going forward with expanding diagnosis?
Well, Mary, this is a really important and practical question. Genetic testing today is quite expensive because it requires specialized equipment and laboratory expertise. And sometimes this equipment and laboratory expertise just simply isn't available in some parts of the world. Measuring ADA2 can potentially be much cheaper and faster, and we can do this in a basic lab using a simple blood sample and does not require advanced technologies.
Well, that sounds a lot like the goal of the ADA2 now meter that the foundation has been working on. So can you tell us a little bit about that first?
Sure. The meter is a small handheld device, looks a lot like a glucometer. We use a few drops of blood obtained through a finger prick, and we envision that this can be done in their doctor's office or in some cases in an emergency room, and we get a result in 15 minutes to determine if the ADA levels were low or normal.
So how does Dr. Lee's research impact the ADA2 now meter?
So if ADA2 blood enzyme reflects a disease biology, then a simple point of care test, such as ADA2 now, would be able to diagnose ADA2 earlier. We know, for example, that there are thousands of undiagnosed patients across the globe, and having a simple, inexpensive device makes it much easier to diagnose patients, especially where genetic screening is just not feasible.
So now the most important question, when will the ADA2 now meter be ready for use?
That's a really important question, Mary, and it really depends, like all projects, on funding. The foundation has applied for a phase 2 SBIR grant from the National Institutes of Health for about $2 million to complete this project. We expect to find out if the grant is approved in the first quarter of 2026. So we're very close to finding out if our funding is approved. If funded, it will take about 2 years to move the ADA2 meter through the rest of the necessary research and the regulatory approval process. If we don't receive the grant, we will continue to advance the project using more traditional fundraising methods.
Well, we're going to all keep our fingers crossed that the grant will come through. Thanks, Lex, for taking the time to explain all this to us.
Great. Thank you, Mary.